Good morning, everyone. Welcome. My name's Jen Goldsack. I'm CEO of the Digital Medicine Society, and I'm absolutely delighted to welcome you to our November webinar, the V3 Framework in Action. I'm thrilled to be joined by my co-authors, my colleagues, my friends that I had the privilege of working on the framework with, Elena Ismailova and Ariel Dowling. But before I introduce them, I'm going to go ahead and introduce DIME to folks on the line who may not be familiar with us. So DIME is a global nonprofit dedicated to advancing the ethical, effective, equitable, and safe use of digital medicine to redefine healthcare and improve lives. We're still a reasonably young organization, founded only in 2019, but we've had outsized impact, for example, through work like the V3 framework, because of how we approach the field. We don't think about digital medicine as being a clinical driven endeavor with consults going out to our tech colleagues, nor do we think about the tail wagging the dog and our tech colleagues driving things forward without true partnership with our clinical colleagues and importantly with patients. So when we think about this model, how do we actually bring these experts to the table? First and foremost, we think about convening interdisciplinary experts ranging from citizen scientists and cybersecurity experts, electrical engineers, physicists, product experts, data scientists, clinical care providers, healthcare executives, payers, regulators, investors, funders, unless we are all speaking the same language. Unless we have the same shared idea of what good looks like, we are always going to be limited. So we build that community first, each of you as a member of that community. Then we think about the research that we need to do in order to advance the field. The V3 framework is a great example of the kind of high impact research that we do when we sit at the intersection of these fields. And then we think about communication and education. And again, today is a great example of that, with leaders in the field like Ariel and Elena coming to the table to tell us how the V3 framework is affecting their day-to-day work and the patients we're all here to serve in action. So delighted to have you with us today and really excited to get all of you situated for a great session. So how is today going to work? If you haven't attended a DIME webinar before, this is how we're going to use our time together for the next hour. So first of all, full disclosure, we are going to record today's session. And while we have expert presentations coming in from our colleagues, we will leave plenty of time for moderated discussion at the end. And so you'll have the opportunity to pose your question live. If you'd like to do that, you can raise your hand, not like this, but in the reactions ribbon at the bottom. If you raise your hand during Q&A, we'll unmute you, bring you to the stage, and you can ask your question to Ariel and Elena directly. If that doesn't work for you for whatever reason, you can pop your question into the chat box. And as I said, we will make sure that we allow a ton of time for these conversations before we break today. And so let's go ahead. And before I ask Ariel and Elena to introduce themselves, and before we dive into the V3 framework, I actually want to take a moment, and I wrote all of our colleagues down because this is important to us. While you have the three of us who are co-authors on the line today, the V3 framework was the result of an extraordinary collaboration. And so I want to acknowledge Andy Korovos, Jesse Backer, Brene Bent, Cheryl Fitzer-Attis, Alan Godfrey, Job Godino, Ninad Gajor, Christine Manta, Barry Peterson, Ben Vandenriesch, Bill Wood, Will Wang, and Jessie Lynn Dunn for this work. This was a true interdisciplinary collaboration. It was a fantastic endeavor. And with that, I feel like I've probably bigged this up enough. Ariel, I'm going to hand it to you to introduce yourself.
Sure. Hi, everyone. My name is Ariel Dowling, and I am the head of Sensing and Measurement at Takeda in the Data Sciences Institute. And my role at Takeda is specifically focused on implementing digital technology and digital devices within the clinical trials. So I work on helping our clinical teams determine if they would like to add a digital endpoint or a digital component to their clinical trial. If so, which one makes the most sense for them to add, and then how they actually go through and do that, how they operationalize it, how they put that in. I come from a background in engineering. So, I spent many years actually working on developing algorithm designs for digital devices and digital technology. And the V3 framework was one of the first paper, maybe the first paper, where I really thought deeply about how what I had done as an algorithm designer could be brought out into a true framework, right? The steps that I had always taken for, how do I feel like my algorithm is ready to be released? How do I feel like my algorithm is ready to go? How do I trust my own algorithm and validate to myself that it's good, right? You know, when I was writing these, I always had to prove to myself first to say, yes, I think it's ready. I think it's done. I think it's robust enough. I think it's ready to do that. And so it was really the first time I actually sat down and thought, well, what are the steps that I take to go through that, and how would that be implemented in a clinical team or in a larger showcase? And you know, as a little spoiler from the very beginning, it wasn't V3 originally, it was V2. And that was it was originally verification and validation. And one of the biggest things that we went through with this entire conversation was, I actually don't think validation as a term is okay. We have to split it because there's two different types. And I think that focus and that split really made it concrete to a lot of people, because I think most people didn't really think about the two types of validation that you do and why, so much so they're both important, but they do serve different purposes. So I think that was, you know, that as a contribution to the field was such a major change in the way people thought about it. And now it's become so much more routine and so much more consistent with how people do it. And now, you know, me at a pharma, I can clearly say, hey, clinical validation is something we need to own. We need to really think about that. And we need to focus on it. We can also do analytical validation, but we're on the hook for the third V, and we need to really focus our effort in our area, in that area.
Ariel, fantastic introduction. And I really appreciate how you talked about the evolution in our thinking as a result of this really excellent collaboration, having these different experts at the table and actually working through that stepwise process was incredibly valuable. And of course, no presentation about technology is complete without a technology challenge. So Elena is having some tech difficulties. So as soon as we get her back on, I will call on her to do her introduction. But otherwise, let me for a moment introduce you all to the V3 framework before we dive in with the meat of our conversation today, which is actually going to be demonstrating the V3 framework in action. But for those of you who might need a refresher, what is the V3 framework? So the really important thing about it is it's a modular evaluation framework. It contemplates each different part of the tech stack that goes into a digital measurement product, and in particular, a sensor measurement product, to allow us to appropriately evaluate whether a digital measurement tool is fit for purpose. So we talk about verification for the sensor. Is the data coming off that sensor correct as compared to a reference standard? Then the analytical validation, how well is the algorithm that's interpreting that sample data from the sensor into something clinically interpretable? How correct is that in the given context of use? So in the environment and in a very particular patient population. Importantly, analytical validation and doing it well is how we ensure that these measures perform equally well, for example, across all skin tones, right? If we're using a PPG sensor, because analytical validation requires that we use a representative sample to test and to develop and test the algorithm with. It also thinks about whether a mobility movement performs equally well in differently abled bodies. This is what's so important about this framework. And then finally, just as Ariel, you said, you need to fully own clinical validation, because clinical validation is something that we've been doing, you know, on the measurement side of the house for decades, long before we even started to think about, you know, what is this data coming off sensors actually mean in practice. And you got to the heart of the matter when you said, you know, this is the section we need to fully own. And one of the other pieces in V3 we said is this is a highly interdisciplinary endeavor. We really need to think about the role and the expertise that all of our different colleagues bring to the table. And so what you see in the V3 framework is not just these are the steps, but this is what good looks like. And this is the documentation that you need to expect at every step. So with that, absolutely proud on behalf of all of the authors to share the impact that the V3 framework has had on the field. So V3, in the two and a half or so years since it's been published, V3 was one of the first projects we worked on here at Dime, been cited over 120 times in the scientific literature. V3 is foundational to how the EMA and NIH think about digital measures. And also the V3 framework aligns really nicely with the recent FDA guidance, the Digital Health Technologies for Remote Data Acquisition, that draft guidance that came out from FDA nearly a year ago now. So V3 really is emerging as the gold standard methodology for developing and evaluating digital measurement products out in the world. And so let's stop talking about it and let's start showing V3 in action. And I'm going to stop sharing my screen, Aria. Actually, that's not stopping sharing my screen, that is, and hand over to you to walk through how your team at Takeda is using different elements of V3.
Alrighty, let me get my screen share up. So we are going to go in a little bit of a different order right now, simply because we're waiting for Elena to come on in. So when she does, we'll be able to do that. So we're going to start with that third V, that clinical validation. And as Jen was saying, in terms of who owns this is really neat. Clinical validation is the measure fit for purpose for a specific patient population in a specific context of use. And so that's exactly why a pharma company or a really major sponsor is the best person to own this, because we're the ones that have access to the patients. We're the ones that, are able to recruit the patients. We're the ones that have the patient data. Also, we're the ones that are trying to understand how the patient feels, functions, and survives, using that FDA criteria. And so while certainly there are most other groups that can also do clinical validation, it's very critical for the pharma sponsors to understand that this really lies with us, and that's where we should be focusing on a lot of our work here. So what does clinical validation of a biomet look like in a major pharma company? So I can tell you about one that Takeda had done. We are going to be publishing this, so while I will be relatively general with the comments today, this will be published hopefully within the next year, and so there will be a publication coming out where we can be a little more specific at what we did. So this was a clinical validation of a portable EEG system, and the study was specifically to study the wake and sleep state transitions on a portable EEG device in narcolepsy patients versus healthy participants. Now, why would we need to do clinical validation in this particular context? The reason is because this portable EEG device had been very well validated and studied in healthy populations and had been shown to have been accurate against the gold standard, a PSG system, polysomnography system, again in healthy patients. However, narcolepsy patients have very different sleep architecture than healthy populations do. And so as a result, we couldn't port that analytical validation that the company had done on healthy participants over to a narcolepsy patient population, because their sleep staging looks very different than a healthy patient does. There's a traditional ordering that a healthy patient goes through of sleep staging, where they go from light sleep to progressively deeper sleep. And so a lot of the staging of the portable EEG relies on the understanding that there is a natural progression of the sleep stages and that you do have to move from one to the other. Narcolepsy changes all that. In patients with narcolepsy, they can actually jump from a very wake state to a very deep sleep state, which would not be possible in a healthy patient. And so that sort of natural transition of sleep staging does not work. And so therefore, we were not confident that the algorithms and the validation that they had done in the healthy population would port over to the patient population. So we felt that we needed to do this on our own in order to understand how this device would work within the narcolepsy population. So this specifically was set up as a validation study, and it was looking for concordance of the sleep state scoring, as well as the correlation of sleep transitions between the gold standard system, an NPSG or polysomnography, and the wearable EEG system. And we did the full validation in the in-clinic portion of the study. So we had two systems they were running, the gold standard and the system under test, while they were in the clinic. The second part was also to understand if we could potentially look for developing a digital biomarker related to the night-to-night variability in sleep patterns of the patients with narcolepsy versus the healthy patients. This was done using at-home data where we only had the portable EEG system. We didn't have the NPSG system, as that's an in-clinic system. And again, this looked at a specific patient population, a specific context of use. This was healthy and narcolepsy patients, both in the clinic and at home. So we used a variety of digital devices in this study, but the main one was a portable EEG system that's lightweight and designed specifically for nighttime use in the home environment. We also added additional exploratory devices to understand more about how these patients were feeling, functioning, and surviving during the study. Specifically, we had a chest ECG system for measuring continuous at-home heart rate and heart rate variability metrics. And we had a wrist-based accelerometry system looking for additional metrics such as activity level, intensity, rest, and sleep. Again, a third and a secondary way of measuring sleep so that we can compare it to the main way of measuring sleep, the EEG system. We also had an ePro system that asks the patients questions about both their sleep quality as well as specific symptoms of narcolepsy. This is the study design. So there's two segments, an inpatient segment and an outpatient segment. The study included 16 patients with narcolepsy and 16 age and gender matched controls. They used the digital devices. The digital devices were the nighttime EEG, the accelerometry, the ECG system, and the ePro. We started out with 2 nights of inpatient testing to enable us to test the EEG device against the gold standard NPSG. So the patients would come into the lab for the first, on day 1. They would have both systems placed on them. They would wear the device prior to bed. They would sleep for the whole night with the NPSG and the device on the first night. The second day was a full day where they were in the clinic. And what we would do is we did a variety of standardized testing for them, specifically an MWT or a maximum wakefulness test to understand exactly, when what the different measurements would show during the standard test. Then they would again do a second night of the NPSG and the device under test. And then we would send them home on day 3. And then from day 3 to day 8, they would wear the device every night on their own, place it on themselves, using it, because again, getting that usability and doing that 5 nights of outpatient testing. And then they would come in on the last day for a debrief. And we also asked them to answer ePros as well to understand their sleep quality and their narcolepsy systems. So 2 contexts of use, one with gold standard, one with additional exploratory devices. So in terms of the expected outcome of this validation study, the goal of this was to really determine if this portable EEG is fit for purpose, really understanding that clinical validation in the specific narcolepsy patient population, with the goal of essentially saying, if it is fit for purpose, how can we use this in our future clinical trials to understand more about how our patients are sleeping at home, in the home environment. Most of the sleep that a person does is going to be in their home environment. There's very rarely they're going to come into a clinic. So the true understanding of this is saying, can we use this device to understand the majority of our patients' sleep when they're in a clinical trial? We also were looking to develop a digital biomarker specifically around sleep transitions. And then our understanding was, can we use this this biomarker to measure at-home metrics to specifically quantify them in terms of understanding how they are progressing within the clinical trial. We also wanted to build expertise with the digital device and build operational experience. By trying this out in a validation study before we put it into a clinical trial, we were building confidence across all the functional groups within Takeda that would potentially work with this device so that they understood how to operationalize it. That's a very underrated experience. Every time you put a digital device in a study, there are going to be some hiccups and special things you need to work with those devices. So being able to trial it in a validation study before we put it into a clinical study really helped us work out a lot of the kinks and understand this is how we would need to operationalize it successfully in a clinical trial. Lastly, the initial results for this were presented at Sleep 2020 by Max Tolkoff, one of the Takeda PIs on this study. Unfortunately, I'm not going to be presenting them here, but we will be publishing them within the next year. So sorry to leave you hanging a little bit, but I do promise that we are working on a publication to get this data out into the world.
Ariel, absolutely fantastic. And it's funny, we talk about V3 being the first project we worked on here at Dime. It wasn't published until the spring of 2020. The joke I have about these things sometimes is moving at the speed of science when it comes to publications. So we'll keep an eye out for that data going live, and really appreciate you walking us through that today. So we started at the top of the tech stack. Elena, you missed it when we were having a little bit of a giggle earlier that no tech webinar is ever complete without some tech difficulties. So really thrilled you could join us. Before we go ahead and ask you to walk us through the application of the verification part of the V3 framework and the analytical validation steps. Elena, it was such a delight working with you, and of course, Ariel and the others on this. Do you want to do a quick introduction before we hand the mic over to you?
Yes, absolutely. Thank you so much for inviting me, and the sentiment about the wonderful experience working on this paper and with the entire Dime community is completely reciprocal. I'm thrilled, and thank you so much for inviting me.
Of course, of course. Elena, do you want to go ahead and share the slides from your computer and walk us through how you and the Koneksa team, your partners, are applying the sort of V3 framework in action?
Absolutely. Let me get to the slide deck in a second.
And if it's easier, I can share from my side as well. Whatever works for you, Elena, is perfect.
The slide deck is loading. Oh, you can share it, Jen. It's not a problem.
No, go ahead. If you've got it, it will be more fun for you to present from your own slide.
Right. I will. Can you see my screen?
Yes, we got you.
Okay, wonderful. So thank you again for inviting me here to share the our experience in working with the V3 framework. And today I would like to share with you our version of V3 in action, which is gait and balance measurements using body-worn accelerometers for patients in Parkinson's disease. And I will walk you through all three steps. As you all know, gait and balance are impaired in Parkinson's disease, and it is possible to measure these impairments by collecting the basic characteristics of gait and balance, such as the walk duration, the walking distance, the number of steps, the walking speed, and stride period. I work for Koneksa. Koneksa is the company that is dedicated to advancement and validation of digital biomarkers and provides the platform for data integration. We are a device agnostic platform. We don't own any hardware. And for this experiment, we used 3 different devices, an iPhone and 2 devices by Actigraph, GT9X and Centerpoint Inside Watch. This is a truly device agnostic approach and helps us leveraging quickly evolving nature of biometric monitoring devices. I would like to stress out here that by measuring gait and balance, by no means we are getting a full picture of all symptoms of Parkinson's disease. This is a very complex medical condition and requires multiple assessments. I simplified this use case for purposes of showing the V3 framework in action. And right now I'm going to walk you through the steps. The first one is sensor verification, as you of course well know this. Yes, Jen.
I'm so sorry to interrupt you. These slides are excellent. Would you mind going to slide share mode so we can see them in their full glory?
Okay. Okay.
Spectacular. Oh, so much easier to see. Thank you.
All right. Excellent. So as you know, the first step is sensor verification. And to understand the accuracy of accelerometers embedded in all these 3 devices I described in the previous slide deck, we put them on top of the shake table, which is gold standard benchtop device for evaluating sensor accuracy. What this table does, it moves with different amplitudes and frequencies that you see in the gray cells in the middle of the slide that result in different accelerations. And we repeated the experiment of going through all these accelerations 4 times. You see the results in the table on the right for each of the device. So the color-coded dots are each experiment, each repeat, and you see the results by interclass correlation coefficient that compares the acceleration recorded by the shake table versus the accelerometer in each of the technology, either iPhone or these wrist-worn devices across different accelerations. So you can see from this picture that for point estimate of accelerations observed close to what we normally expect during walk, the agreement according to the ACC, it's good or it's excellent because most of values are above 0.8. So all three devices, the accelerometers in all three devices are fit for purpose to record gait and balance. But I have another data set on a lower part of this table. And for instance, if we wanted to look at accelerations normally observed during hand tremor, another feature of Parkinson's disease, the accuracies are different. The iPhone is performing similarly across all accelerations, but not GT9X and CPIW. I would like to emphasize that this doesn't mean that there's something wrong with GT9X and CPIW. Not at all. These are very good devices, and we work a lot with them. These are 510K cleared devices by the FDA with intended use of collecting physical activity and sleep data. For this purpose, the accuracy at lower accelerations is not important, and that's why we see what we see. But if we didn't do this verification and we wanted to use these devices for collecting the hand tremor, it shows that this solution is not optimal and requires some adjustment of how the accelerometer records the data. What we learned from this experiment that the verification has to be run regardless of the regulatory status of the technology, and context of use is of an absolute importance. We were able to do these experiments because the device manufacturers give us access to sample level data. It doesn't happen all the time, and it's not available for all devices. So the devices where the manufacturers don't give access to the data will be difficult to assess according to the V3 framework, and in some cases it can be done only by the technology developer. Now I'm going to switch gear and talk about analytical validation. We did analytical validation in healthy volunteers, and here I'm showing the data from validation of iPhone. We used as a benchmark the raters done as a benchmark, the rating done by trained raters for duration, distance, and steps. Speed and stride period were secondary derivatives to which we compared the output of the algorithm. The study subjects walked for different durations, and they kept the iPhone in a tight pocket. We also tested for operational tolerance, which is by placing iPhone in a loose pocket or in a shoulder bag. And again, we used interclass correlation coefficients shown in this table, which shows that they were good or excellent for most of the testing conditions. I also would like to discuss a little bit of doing analytical validation in healthy volunteers versus disease population, because in this case, it was possible to do it in healthy volunteers because gait and balance features can be recorded in both populations. And in many cases, it's an optimal first step because testing novel algorithms, and here I'm talking about algorithms developed by Koneksa that may require multiple iterations, which would better be done in healthy subjects. However, if we are talking about hand tremor, this needs to be done in Parkinson's patients because healthy people don't have appreciable tremor. And this applies to any algorithm, newly developed or publicly available, because the context of use is very important. The algorithm may be available publicly, but it may have been developed for a different purpose, and that's why it needs to be tested in the context of use. Recently, we published our study in the Sensor Journal, and this was an interesting experience because we referred to V3 framework, and what I learned going through the peer review process is that V3 framework not only guides the community how to design and execute experiment, it is transformational for educating of our community because it helps building a tribal language. When we talk about fit for purpose concept and what needs to be done, we are all speaking the same language. And by a tribe, I mean here us, everyone who works in the digital medicine society. And we were able to easily to explain to the reviewers why V1 in V2 are important and why they need to be done first in the healthy volunteers. In the absence of the V3 framework, it would be much harder to do because a lot of people jump immediately into the clinical validation because it contains an ultimate answer. And very often the V1 and V2 data is not available, which brings me to the point of the final step, V3. And I'm not sharing any data here because the study is still ongoing, but it's wrapping up as we're speaking. What we are doing in the clinical validation, we are comparing the outputs of the from the accelerometers and algorithms to the benchmark, which is MDS-UPDRS, a clinician rating scale. And this study is, of course, done in Parkinson's patients. We are exploring the relationship between MDS-UPDRS, construct validity, sensitivity to change, and test-retest reliability, comparing it to corresponding items of MDS-UPDRS. However, we are doing additional features, which is usability. Can patients use the data? Can they use it safely? Can sites deploy these technologies successfully? And at the end of the day, if data makes sense. These features are not in the V3 framework, but I would like to inform you that DIME works really hard in addressing other questions around what will make a complete package validating certain biomat to be fit for purpose. And I'm happy to announce that there will be another DIME exciting project dedicated to usability. Please stay tuned and check the DIME website. Thank you very much.
Elena, magnificent. We're getting some really good questions coming in. Appreciate you and Ariel really sharing how this works in practice. I think some of the comments you made were incredibly important. The fact that, sometimes we are inclined to sit where we're most comfortable, especially on the clinical side of the house. So we go straight to that clinical validation step and we skip over these other steps. Through work we did on the playbook, we actually identified through what we describe as a pre-mortem activity, which with the benefit of hindsight, I wish we called something different. But we said one of the greatest risks is actually not taking the time for the new and critical steps. Things like the analytical validation, making sure that we are checking that sensor performance through verification, but then that algorithmic performance in the correct context of use before moving ahead, doing the clinical validation and deploying. So such an excellent point. Before we throw it open to questions, Ariel, I might ask you just to close us out, to sort of summarize from your perspective how the V3 framework comes together and influences the field.
Sure. So I think that the biggest thing is that this is a modular framework. It's just a modular evaluation of the digital measures. It includes those 3 steps, verification, and then the 2 forms of validation, analytical and clinical. And as we said specifically, they do have very different meanings. The analytical validation is when you test your algorithm performance against the gold standard. So Elena showed a really great example of that type of validation. And then the clinical validation is, what does the DHT accurately measure for a meaningful outcome metric for a very specific patient population? And so I showed that with the portable EEG system that we were looking for to measure sleep parameters, specifically for patients with narcolepsy. Really, the whole goal of the V3 framework, and the reason you go through it, is because you're trying to answer the question is your measure, is your health technology, is your biomarker, is what you're trying to measure fit for purpose? And if you go through these 3 steps, you'll be able to definitively show that yes, it is fit for purpose for what you need to do. As Elena said, it's not the end. It's not the end. There are other steps to do. There are things beyond clinical validation. There's data privacy, security, et cetera. But if you're specifically looking to answer the question, or if someone asks you the question, is this fit for purpose for my study or for my patient population or for how I want to use it? You can say, yes, I've gone through these steps and this is how I've shown to you that this is how it is fit for purpose. And that way it gives you a way to answer that question. Whereas before it was kind of all, I would say everybody was sort of left to their own devices. Now we've defined a definitive framework to say, this will help you answer this question. And you can refer back to it to say, this is what the field has recommended for this type of analysis.
Spectacular. Excellent. I think that's really important. Stressing that this is a modular framework is really important. And the reason I also remain very proud of the modular approach that we took, and that I truly believe is sort of fit for purpose for evaluating these sorts of things, is it also allows us to decouple the different parts of the tech stack, right? We can select the actual measurement tool, the sensor product that's generating sample data that is the best fit for a given patient population. And in different contexts, we may want to switch out that sensor, right? Algorithms improve over time. We may want to translate a measure into a different clinical context, right? We can think about these different areas. That's the power of this. It not only is rigorous evaluation science, but it also helps us think about being as effective and efficient as possible in developing and translating these tools and measures into different contexts. So we've got excellent questions coming in. Alan Godfrey had a really good one that came through the chat before we move to the Q&A. Alan, I've unmuted you from my side. If you'd like to unmute yourself and ask your question live, please do.
Oh, hi, Jen. Really good by Elena and Ariel. Hope you can hear me. I just want to say from my own accord, it was a wonderful experience to work with everybody on this paper as well. And I can just say from my own academic research, it is something that I do dip into quite often. It has been a very, very useful platform to springboard into some of the technology and algorithm development work that we do here as well. So from my perspective, it was a wonderful learning experience and a very, very useful resource to utilize my ongoing academic work. So thank you, Jen, for initially inviting me to the party a number of years ago, and for Elena and Ariel giving a wonderful presentation on the effectiveness of the V3 framework. Sorry, I've gone off piste a little bit. It was a very short question to Elena, just to briefly ask Elena, what was the sampling frequency that you had set with the GT9X when it came to the hand tremor study that you did?
Alan, I would have to get back to you because it's not in the specific experiments and paper. I used it in the illustrative example. Right now, I don't have the number off the top of my head. I need to get back to you on that.
Okay, that's no problem. Thanks, Elena.
Mm-hmm.
Fantastic. And Al, really good to hear your voice. It was a pleasure working with you on this, and glad you could join us today. And speaking of folks who joined us on the project, Cheryl, delighted to have you on the line too. I'm going to, I don't seem to be able to unmute you. Let me see if I can try this one more time. And if I can't, I'll go ahead and ask you your question on your behalf. Yes, I've unmuted you on my side. Cheryl, do you want to pose your question live?
Hi, Jen. Hi, everyone. Yeah, I echo what's been said. It was a really it's a fantastic experience writing that work virtually, right before the pandemic. That was probably the first manuscript I did in such a manner. My question was to, like we heard Ariel, we heard a little bit not in the order that was intended, but we heard from Ariel about the importance of doing the clinical validation in the narcolepsy population as opposed to the healthy volunteers, and that makes a lot of sense because the difference in the sleep architecture. Elena, you didn't hear that comment. I'm sure you were aware of that, but you brought to us a validation that you did for Parkinson's disease, for gait and posture balance for Parkinson's disease patients eventually, but you did the validation in healthy volunteers. So that seemed to be sort of a disconnect there in the message that was coming across, and I wanted to understand or ask you to elaborate on that. Because certainly posture and gait and balance are different. There's a different architecture to that in Parkinson's patients versus healthy volunteers.
This is a really good point, Cheryl. Thank you for bringing this up. Yes, you're absolutely correct that it needs to be tested in Parkinson's patients. There's no question about it. So I would like to emphasize, and maybe I didn't make it very clear during the presentation, that testing in normal healthy volunteers was the first step of analytical validation to make sure that algorithm is working properly before putting it in the disease patients, because as I said before, sometimes optimizing the algorithm and making sure it's working properly requires iterations, and this is best to be done in healthy people before putting it in the disease population, because these people have disabilities, and asking them too much repetitive work, in my opinion, is not ethical to jump immediately in the disease population before the tool has been optimized. I hope I have answered your question.
Absolutely. Thank you, Elena. Absolutely.
Fantastic. Thanks, Cheryl. Appreciate that. And Elena, terrific answer. I'm going to come, Caitlin, I'm going to see if I can unmute you for our next question. Give me 1 second here. Caitlin, if you'd like to unmute yourself, please go ahead and pose your question live.
Thank you very much, Jennifer. So I had a question about the difference between analytic and clinical validation. And just, I think as some background, I work within our patient-centered outcomes research team within AbbVie. So definitely kind of part of the group with psychometric validation expertise and measurement expertise in that sense for clinical validation. But I think sometimes I get confused about analytic validation, particularly for certain therapeutics where it seems that there are certain parts of analytic validation that essentially are clinical validation. So I'm just curious, are there more granular parts of analytic validation that we could kind of specify, or kind of what parts of analytic validation specifically requires folks with digital health systems and operational expertise and kind of the technologic expertise that's needed? That'd be really helpful to understand.
Yeah, so I think that, you know, with an example like the wearable EEG study, right? The verification was, are we actually getting EEG measurements out of this? Is this an EEG signal that comes out of it? The analytical validation is, does it accurately measure sleep at all? Can it measure sleep? And then the clinical validation is, does it measure sleep in the specific patient population of interest? That's not as clear if you're looking at an algorithm that specifically is looking at a patient symptom or a patient phenomenon. So let's say you're trying to measure bradykinesia in Parkinson's disease, right? You have to decide how you're going to measure bradykinesia. Most people do it using a movement sensor, right? So your verification would be, is the accelerometer and gyroscope putting out accurate measurements for measuring the bradykinesia? Your analytical validation is going to look a little different now. Basically, you're saying, am I measuring bradykinesia, right? So your analytical validation does have to be done on Parkinson's disease patients, because there are, you know, healthy patients will not have bradykinesia. So you do have to do it. But the analytical validation is what I'm measuring when I measure the sensor bradykinesia, or is it just regular movement, right? How do I split between normal everyday hand movement of the patient and actual bradykinesia if you're putting it on the wrist or on the legs or anything? So your analytical measurement is the algorithm I have putting out the metric that I think it is. And it may be done on patients. Now, the clinical validation will look a little different. And that question will be, does this matter to patients? Like, does this, you know, for bradykinesia, is bradykinesia an important measurement for patients, things like that. So because of that, you may do them both in the same study because you're looking specifically at Parkinson's disease palliation. It doesn't necessarily have to be two separate studies or two separate steps, particularly when you're looking for a patient phenomenon. You may be able to do a single study for it, but you do have to have two different aims. The first one is, am I actually measuring bradykinesia? And the second aim is, does this measurement matter to the patient in the specific context of use and in this specific patient population? So that's why those two, they are separate, but they may be together in a single study, and you may be able to answer them in a single study, which would happen if you're looking at a very specific patient phenomenon or very specific biomet that comes from a patient. But the questions are still distinctly different. One is, can you measure it? And the second is, why should I care that I can measure it? Because not everything you can measure matters.
Okay. Yeah, thank you. That's really helpful. Are there situations where you found that they are kind of one and the same, like the gold standard we're comparing against to say, are we measuring bradykinesia or any other example is the same as how we assess what matters to patients? Or does that not happen very often in your experience? That hasn't happened?
I think I got that. There was a little background noise there. So in terms of what matters to patients, that's where the FDA framework of how they feels, functions, or survives really comes in. For bradykinesia, does that affect how the patient feels, functions, or survives? Most Parkinson's patients will certainly say yes to that. They feel that bradykinesia is something that does affect their daily life. It does affect how they generally live. This is important to them. And so you may not have to prove that bradykinesia matters to PD patients, because there's a large body of literature that can support that. But if you're working in a rare disease, so at Takeda we work in a lot of rare diseases, this may not be obvious to the health authorities, right? It may not be obvious that this particular measure matters to how the patient feels, functions, or survives, simply because it's a very rare disease and it's not as well characterized with the patient population. With the large ones like Parkinson's disease, you can rely on additional literature, and you may not have to run additional work on that. You could do, usability studies in there. You could, do patient populations, talking to KOLs, but the crux of your actual study validation may focus on that analytical validation of, am I measuring bradykinesia when I say I measure bradykinesia? Because you're building on a large body of literature that already says this is clinically valid for our patients, but that won't be the case for every metric. I'm trying to think of an example of one where it's not as well established. I think, in Duchenne's muscular dystrophy, the EMA has approved a single digital endpoint on it. That's the 95th centile of stride velocity as a registrational endpoint. So that company had to prove both that they can measure 95th centile stride velocity in DMD patients, and also that this measure mattered, right? That this metric was important to the patients, and that it was something that affected how they feel, function, or survive. So that was not an obvious one that they had to do. I believe that they did that combined together in a single study, though please don't quote me on that. I'd have to go back and look on the paper, but I believe they did that as a one combined study, is the analytical validation as well as the clinical validation together. Again, that's more specific to when you're looking at a very specific patient population. And again, on a rare disease like DMD, you would need to prove that as well. So I think that when a pharma company, I believe you said you work for AbbVie, is doing that, when a pharma company is doing it, there may be some times where you can sort of squish the two together to say, yes, we can do them together. But it really has to be evaluated on a case-by-case basis. And you have to think about how you're answering both of those questions over the course of the study.
Thank you.
And Ariel, you made a few really good points there. And I want to reiterate what you said about, just because you can measure something, you can have a beautifully performing algorithm. You can be measuring something absolutely correctly if it has no bearing on that patient population. And that's the difference between your sort of analytical validation and your clinical validation. then there simply isn't value there to any stakeholder, the patients themselves, the researchers or clinicians trying to make decisions based on this measure, and then downstream decision makers like our regulatory colleagues or even our payer colleagues. I think that, Caitlin, it's such a great question, and I'm so glad you asked. Taking the time to distinguish between the two different questions that analytical validation and clinical validation ask stop us from spending our time measuring things that don't matter to a given patient population. And, the whole, just because you can doesn't mean you should, I think probably applies really well here. Pip, I'm going to go ahead and unmute you for our next question. Give me 1 second and you should be able to talk. Pip, you should be able to unmute yourself from your side now.
Testing.
Yep, we got you.
Great. Okay. Yeah. So earlier I was having a chat with some of my colleagues and collaborators, and we were talking about this idea of gold standards. And today I said in my question about using gold standards for clinical validation, but also that you can use them for analytical validation. We talked a bit about that today. But let's just think about, I think in the examples that you showed, you were comparing against the MDS UPDRS, for example, to say, okay, well, let's say that's our gold standard, and we can compare our device against it. Now, the problem is, I think we're in an era where our tools are probably better than the gold standards. If we just take a very simple example, like a single item from the MDS UPDRS, which is like 4 categories or 5 categories, I can't remember, against some really high-tech continuous measure, there's just going to be less information in a single item. When we correlate those two things together, hopefully we're going to get quite a nice correlation. But there's always, it's never going to be perfect, because you never would expect it to be. So there's some error of residual or noise. But is this noise really noise? Or is the deviation, the residual from our gold standard, actually that signal? That's important. We should be trying to capture that and say, hey, look, any deviation from our gold standard doesn't mean that our measure is performing poorly. It actually, we need to try and understand what that noise is. So my question there is, what are your thoughts on comparing against gold standards, where we are now, and how to combat that?
Elena, do you want to take this one?
Certainly. This is a really important point and has been discussed at every single conference on digital measures that I remember. And how we select gold standards, if indeed they are gold, because your point, Pip, is well taken, that it's a limited scale with that we are comparing very granular measure to. But in my take on this, that at this time we need to start with using these gold standards, because this is a paradigm for clinical validation. We are comparing it to an established outcome of interest that happened to be a rating scale, and comparing it to a single item of a clinician rating scale has certain limitations, no question about it. Looking forward, my prediction is that there will be another different methods, and we will do it differently in the future as we have more measures and related endpoints approved and being used. And the example that Ariel brought up about 95th percentile of Duchenne muscular dystrophy at some point may become a gold standard, and it is already a digitally derived measure, and probably comparing to something like 95th percentile of stride velocity will address some of the challenges that currently we are facing. Ariel, do you want to add anything?
Yeah, I think that, this is always the challenge, right? If you tie yourself to a specific gold standard, you can never be better than the gold standard, right? You're never going to beat it, because you can only show, oh, I'm equal to it. I can't show better. So the selection of gold standard then really matters. And it may not be a single point, right? It may not be a scale, right? It may be a combination of things. You may say, all right, we're going to look at the general trend that looks at it. Does it go with the same trend as the scale, but do I see a steeper slope? You know, it doesn't, when you select a gold standard, you don't necessarily have to tie yourself to a single gold standard, right? Or it can be a combination, you know, essentially like a Z-score of a variety of different things. The goal here is you need to convince yourself enough that it's working and have enough data to back it up. So if your gold standard is not so great, maybe it's more of a silver or a bronze standard, you may want to pull in things like, you know, patient-reported outcomes, which certainly can be used as a comparator. You want to pull in a variety of different things to show, yes, we're tracking with general disease progression, or we're tracking with the general timeline, or things like that, because there is in a lot of places, there's not going to be anything you can pull from. You really need to think about how you demonstrate that this matters as it feels, functions, or survives. But a gold standard doesn't have to be a single device or a single measurement or a single pro. It may be a combination of them. It may be a combination of a few disparate items. It may be comparison to just general natural history progression. It may be comparison to previously recorded data. It's really up to you to justify the use of a specific gold standard, and one that will make you feel confident that your measurement is going to go beyond that. Because as you said, a lot of our tools are more sensitive now. They are more specialized, and we may be able to beat what's currently out there. So we don't want to hamstring ourselves by tying ourselves specifically to a particular measure that may not be as relevant now. But it's definitely it's more of an art than a science, I would say, in choosing the right gold standard when there isn't a clear example. For us with the EEG device, we had a very clear example. That was an easy one. We didn't have to really think too hard about it. But especially when the standard is a scale, right? And I know the UPDRS is one of the most challenging ones. It's very, very hard to decide how you're going to demonstrate efficacy beyond that. And it's really, really takes a lot of thought into what you're trying to measure and how you can demonstrate that it's more effective than the current standard.
Terrific answers in both cases. And I do think this idea of, you know, and we talk about it a lot in the paper, being really thoughtful about what that reference standard is. And indeed, considering whether you actually have to use, just like you said, Ariel, that sort of basket of references to make your case, to prove it out, is a really good approach. Let me go ahead. I know there's a couple of questions we didn't have a chance to get to, so I invite all of you to come across, jump into the Dime Slack, and hopefully we can continue the conversation there. We'd be delighted to do so. And then as we round out today, and before I thank you properly, Ariel and Elena, I do want to say for folks on the line who are actively using V3, we warmly invite you to let us know. We'd love to spotlight your work in action through our Resources in Action Hub. And then Elena, as you alluded to, and as we've talked about a lot today, V3 is an extraordinarily important framework. It helps you determine the measurement performance and whether it is fit for purpose for your given patient population. And there are more questions that we need to answer. So we're thrilled to be able to share that we are extending our V3 framework through a new project that will launch in Q1 of next year, to really consider principles of human factors, human-centered design and usability, and make sure we're contemplating not just whether the measurement tool performs in quite controlled situations, but whether you can capture during activities of daily living in the real world, the highest quality data possible. So warm invitation to join us. Ariel, Elena, thank you so much for being wonderful collaborators in this work. It's fun getting part of the gang back together, and it was great seeing some of our co-authors on the line today. I am incredibly proud of the impact that this work has had on the field. And for folks on the line who may want to join some of our upcoming programming, our next webinar is on November 30th, and our DE&I and digitized clinical trials team will be joining us to talk about how we can ensure inclusion in digitized clinical trials. Just a day later, we'll then have our virtual first team. So everyone on the care side trying to build care around the patient, not the clinic, will be reporting data that we actually launched and announced today in partnership with our colleagues at Omada. We completed the first benchmarking survey of the use of virtual first care here in the US. And then finally, last but not least, our next Journal Club on December 7th, our own Pip Griffiths will be leading a conversation around her fantastic paper with co-author Diana Rofail, The Patient Matters in the Endpoint. And I cannot think of a better way to wrap up our conversation than with that particular nugget. So Ariel, Elena, thank you again. Thank you everyone on the line. This is a real pleasure.
Thank you, Jen. It's always great speaking with you.
Thank you. Thank you for inviting us, and let's take a moment to celebrate what we have achieved.
Absolutely. Moving things forward together. All right, everyone, have a great day.